In this video, we will go over the Cytosplore Simian Viewer system demonstrating two use cases. We begin with the startup screen where we see 5 different views. The heat map view displays the in-species cluster distances between two species. The two scatterplot views displays the low-dimensional embeddings of the cells specific to each species and are colored by their cross-species cluster colors. The differential expression view displays the cluster-specific mean expression values for each species along with their differential expression value and other gene-related attributes. And the expression comparison view displays the mean expression values for all the species for multi-species comparison. We now want to make comparisons between human and chimp in the neighborhood non-neuronal cells. The x-axis of the heat map represents the in-species clusters of human and the y-axis represents the in-species clusters of chimp. Each of the small blocks represents the distance between the in-species clusters. Several small blocks are further grouped into cross-species cluster blocks. When we hover over the in-species cluster block, a tooltip appears showing the cluster-related information for both the species. The heat map also shows the cell counts for each in-species cluster block at the top for the x-axis and at the right for the y-axis. Next, for our analysis, we first focus on the heat map view. We find that the OPC1 and OPC2 clusters have very low distance values. We also see that in the OPC1 cluster, the layer distribution of cells are different in chimps than humans. We see that some cells are present in layer 1 and layer 4 for chimps, which is not the case for humans. We now want to further investigate the layer distribution for this cluster in gorillas in comparison to humans. We therefore select gorilla and navigate to the OPC1 cluster. We find that for the OPC1 cluster, gorilla also has some cells present in layer 1, which is different from humans. This is one of the findings that our collaborators have identified in their study. Next, we again compare between human and chimp. Based on the cell count bars at the top and the right, we see that the oligo-1 cross-species cluster block has the maximum number of cells present. We select the cross-species cluster block for further exploration. We can also explore the cell distribution in the scatterplot views where the oligo-1 cells are highlighted in blue. Interestingly, the cluster also consists of low cluster distance values as represented by the dark colors of the block. Therefore, we want to further explore this cross-species cluster block on a gene level. In the differential expression view, we find that the table is sorted based on the positive maximum differential expression values. We scroll down to identify highly differentially expressed gene with a H-condal value associated with it. We find that the LAMA2 gene has a H-condal value associated with it. We also see that it has a high expression value in human than chimp. We want to further investigate by comparing the expression values of all the species for this gene. So, we select the gene and compare the expression values for all the species in the expression comparison view. We find that the gene has a low expression value for all the other primates compared to humans. Our collaborators also mentioned this gene to be differentially expressed in humans for the oligodendrocyte cluster. Thank you for watching. In case you have any questions or feedback, please reach out to us through our website.